Hi, my name is Raja Cohen, and my senior project is a research review of the effect of chronic stress on bacterial populations of the gut microbiome. I chose this topic because the connection between GI health and overall health interests me, and I wanted to take this opportunity to learn more about this interest in a way that will be applicable to my future high-stress career path. For some background, the gut microbiome is a complex network of trillions of microorganisms inhabiting the human intestinal tract. Fun fact, there are more bacterial cells in your body than human cells because of the vast abundance of the gut microbiome. It plays a known role in digestion, metabolism, immunomodulation, endocrine and nervous system regulation, integrity of the intestinal mucosa, and mental health of the host. Stress is defined as a state of worry or mental tension caused by a difficult situation. It is a normal response to everyday challenges, but can have negative effects in high amounts and when experienced long term. It is also one of the many factors that can alter the composition of the gut microbiome. It is important to understand what specific alterations occur in the gut microbiota as a result of chronic stress to better understand its effects on human health and well-being and develop tools to combat any potential negative effects. Here are some key findings from this review. Results from 3 of the studies reviewed identified a decrease in overall diversity and richness of the gut microbiome, and a 4th study observed a decrease in only beta diversity of the microbiome. All 5 of the in vivo studies reviewed observed significant changes in the abundance of bacterial populations following stress exposure. A significant increase was observed in phyla Pseudomonodonta, and a significant decrease was observed in phyla Actinobacteria and Verruco microbiota. Contradictory results were observed regarding the 2 main phyla of the human microbiome, Firmicutes and Bacteroidetes. Contradictory findings were also observed in phylum Bacteroidea. Strength of the studies included in this review are the use of controls for potential confounding variables, for example, the use of both an overall control group and a food and water deprivation control group when the experimental group was deprived of food and water. All studies reviewed provided the same light and dark ratio, food and water, to the subjects, and high-quality statistical analysis was used that took into account confounding variables. Weaknesses of this review as a whole is that it only included in vivo studies performed in mice, which limits the applicability to implications for human health. Weaknesses of the study analyses include a short study duration, which can decrease reliability of results, and failure to address the mode of determination of sample size, which indicates a potential for large margin of error. Based on the results showing significant changes to the gut microbiome as a result of chronic stress, I would recommend developing stress management skills. These can include meditation and other relaxation practices such as yoga, taking alone time to decompress, or talking through stress with friends and family. Other practices to help reduce stress are to engage in regular physical activity, consume a healthful and balanced diet, and get adequate sleep every night. Potential future research in vivo studies could be done to address contradictory results observed in this review, to include a wider variety of phyla, and to address changes at other taxonomic levels, such as the family, genus, and species level. Potential future research could also be done in humans to better understand how chronic stress affects specifically the human gut microbiome, and to understand how these changes affect human health. This could be done ethically through a cross-sectional intervention study, where the intervention is the application of stress management rather than the application of stress itself, or a cohort study following 2 separate groups, one with high stress lifestyles and one with lower stress lifestyles. Here are a list of my references used for this presentation. Thank you so much for listening.